Assalamu alaikum dear students. I am Muhammad Rauf, lecturer, Department of Zoology Cust. Welcome to the course Principle in Animal Life 2. This is lecture number 23. The topic is structure of DNA. Discussing agenda for the topic is Introduction to DNA, abbreviations of DNA, basic building blocks of DNA, comparison of DNA and RNA nucleotides, nucleotide polymerization via phosphodiester linkage, breaking down the structure of DNA, dimensions of DNA, experiments reveal that DNA is the genetic material, DNA is a double-stranded helix, structure of RNA. Lecture outcome. After watching and listening this lecture, students will know about the chemical structure of DNA, its individual building blocks, historical perspective of the elucidation of structure of DNA, and differences with its molecular cousin RNA. Introduction to DNA. DNA is the hereditary material and it carries information for the development of an organism and for the synthesis of enzymes, proteins. Zygote is a single cell and the single celled zygote develops into a complete organism, and all the instructions for the development of a particular body plan lies within DNA. Similarly, hundreds of thousands of enzymes works in our bodies and they run biochemical reactions, and all those enzymes are encoded by DNA. DNA is a double-stranded helical macromolecule. DNA is a polymer composed of building blocks called nucleotides. Human DNA is composed of 6 billion nucleotides and is 1.5 to 2 meter in length. The DNA is located in the nucleus of a eukaryotic cell. Mitochondria and chloroplast have their own DNA molecules. Why DNA is named so? The full name of DNA is deoxyribonucleic acid, and it can be dissected into three parts. Deoxyribo refers to its form of the sugar. The sugar which composes DNA lacks an oxygen at position 2 of the pentose sugar, so therefore it is called deoxyribose. Nucleic, because DNA is located in the nuclei of eukaryotic cells, and acid because the phosphate group is in the ionized form after donating a hydrogen. So the name deoxyribonucleic acid represent some of the characteristics of this molecule. Basic building blocks of DNA. Nucleotides are the monomers. These are the basic building blocks of the polymer DNA. Each nucleotide is composed of three components. Now nucleotides are the building blocks, and nucleotide itself is composed of three components. This one is the pentose sugar, this is the phosphate group, and the nitrogenous base. The nitrogenous bases present in DNA are of four types and broadly categorized into two types on the basis of their structure. These two are double-ringed nitrogenous bases and they are called purines, whereas these two are single-ringed and they are called pyrimidines. Comparison of DNA and RNA nucleotide. As we stated in the beginning of the lecture that RNA is the molecular cousin of DNA. So let's have a look at the nucleotide composition of both these nucleic acids. Thymine and cytosine are single-ring structures called pyrimidines, whereas adenine and guanine are larger double-ring structures called purines. Here you can see once more adenine and guanine are double-ringed and they are called purines. Thymine and cytosine are single-ring, they are called pyrimidines. This is a DNA nucleotide. DNA is composed of this particular type of nucleotides, whereas the molecular cousin of DNA or the RNA is composed of that particular nucleotide. Now, let's come to the differences between them. The DNA nucleotide can be part of any of those four bases: adenine, thymine, guanine, and cytosine. Whereas one difference lies here. Uracil is present instead of thymine in RNA. RNA lacks thymine and DNA lacks uracil. This is the first difference. The second difference is which determines the longevity of the molecule is that position 2 of the pentose sugar. You can see here that the DNA nucleotide has no oxygen at position 2 of the pentose sugar. This is pentose sugar 5 carbon. This is the pentose sugar of RNA nucleotide, and you can see here an extra oxygen is present. So the first difference is the presence of uracil instead of thymine in RNA, and the second difference is the presence of an extra oxygen at position 2 of pentose sugar in RNA, whereas extra oxygen is absent in DNA at position 2 of pentose sugar. So that's why this deoxyribo signify the absence of oxygen at that position. Now, in the Zoology 1 course, you have already studied the four types of biological macromolecules in detail, and you also know that how a monomer gives rise to a polymer. What type of chemical reaction is involved? That reaction is called condensation reaction or dehydration reaction. Condensation reaction is one where the monomers come together and they form a polymer. So here you can see the two nucleotides. They form hydrogen bond with each other, and in this reaction a water molecule is released and a phosphodiester linkage is established. The bond between phosphate group and pentose sugar in DNA is called phosphodiester linkage. It's a condensation reaction. A water molecule is released. Now we further investigate the structure of DNA. Here in the two images, only one strand of DNA is shown. Here you can see the phosphate forming phosphodiester linkage with the pentose sugar of another nucleotide, and similar type of phosphodiester linkage takes place. Now the DNA is a double-stranded helical macromolecule. There are two strands of DNA, and the two strands are arranged or oriented in opposite direction. One strand runs from 3' to 5'. The other strand runs from other direction to 3' to 5'. Now, why they are called as 3' to 5'? It is because of this pentose sugar and the phosphate group. You can see here in that 3 to 5 strand, the third position is below, the third position is below, and the phosphate, which is the fifth position, is above. In the other strand, you can see the third position, the hydroxyl group, is above, and the fifth position, the where the phosphate group lies, is below. So that's the reason why the two strands of the DNA are oriented in opposite direction. Means they are anti-parallel to each other. Now we have discussed one big characteristic of DNA, which is that they are anti-parallel to each other. Now we discuss one another characteristic of DNA, that is its two the type of hydrogen bonding that exists here. Guanine can only form bond with cytosine, and thymine can only form bond with adenine. This is called complementarity. So if you know this principle of complementarity and you have a single-stranded DNA, then you can draw its other strand on the basis of this complementarity principle. Coming to the dimensions of DNA, here the values have been shown in two different units. One is nanometer, the other is angstrom. 1 nanometer is equal to 10 angstrom. So the diameter of DNA is 2 nanometer or 20 angstrom. What is the distance between two consecutive bases in an upright? It is 0.34 nanometer or 3.4 angstrom. DNA takes one complete turn after every 10 nucleotides. One nucleotide is twisted at a 36-degree angle from the first one, and the third nucleotide will be twisted at 36 degrees from the fourth one, from the second one, and so on. So a complete turn is completed after 10 bases, and the distance covered in one complete turn is 3.4 nanometer or 34 angstrom. These are the uprights of the DNA. The uprights of the DNA, and these are the rungs. So the uprights are made up of sugar and phosphate, and the rungs are made up of nitrogenous bases. Experiments reveal that DNA is the genetic material. We have already discussed those experiments. There were 3 different sets of experiments performed, which ultimately proved as an evidence for DNA to be designated as the genetic material. DNA is a double-stranded helix structure determines the function. This is a very important rule in biology. And in biology, you will see lots of examples of this particular phrase. So after the revelation of DNA as the genetic material, the scientists started to investigate the structure of this molecule, because without knowing the details of its structure, they would have had no idea about the function it performs and the way it performs different functions, the way it codes for enzymes and proteins, the way it directs embryonic development. So in order to know about the function of DNA, it was necessary to find out its structure. After Hershey-Chase experiment, most biologists convinced that DNA was the material that stored genetic information. A race was on to determine how the structure of this molecule could account for its role in heredity. Researchers focused on discovering the three-dimensional shape of DNA. Now there were 2 groups of scientists, Watson and Crick on one side, and Rosalind Franklin and Morris Wilkins on the other side. They were working independently to elucidate the 3D structure of DNA. Watson and Crick's partnership solved the puzzle of DNA structure. While visiting the laboratory of Morris Wilkins at King's College in London, Watson saw an X-ray image of DNA produced by Wilkins' colleague Rosalind Franklin. Rosalind Franklin, along with Morris Wilkins, was working through a technique called X-ray crystallography to reveal the structure of DNA. And one day, Watson visited his lab, visited their lab, and Morris Wilkins, the colleague of Rosalind Franklin, showed Watson the photographic image they had obtained through X-ray crystallography studies. A careful study of the image enabled Watson to deduce the basic shape of DNA to be a helix spiral with a uniform diameter and the nitrogenous bases located above one another like a stack of dinner plates. We have already seen in the previous slides where how the nitrogenous bases are arranged one upon each other, just the same way you stack dinner plates one upon the other. The thickness of the helix suggested that it was made up of two polynucleotide strands. DNA, as you know already, is a double helix structure and it has two strands. The two strands are oriented in opposite direction. So initially Watson deduced the information from that particular picture that DNA is double helix spiral, uniform diameter, and the two polynucleotide strands, nitrogenous bases stacked one upon each other. So this was the deduction of Watson after observing the Rosalind Franklin's X-ray image of DNA. But how were the nucleotides arranged in the double helix? Watson and Crick began trying to construct a wire model of a double helix that would conform both to Franklin's data and to what was then known about the chemistry of DNA. Watson and Crick didn't perform any wet lab activity. They did not perform any experiment, but these were intelligent in a way that they carefully studied the work of other people and then they tried to give a practical shape to the findings of other researchers. That was where their genius lied. They knew that Franklin had concluded that the sugar phosphate backbone must be on the outside of the double helix. We already discussed that the uprights are made up of sugar and phosphate, forcing the nitrogen bases to swivel to the interior of the molecule. But how were the bases arranged in the interior of the double helix? Nowadays, we know that the bases are arranged on the basis of complementarity. Adenine always forms bond with thymine, cytosine always form bond with guanine. But at that time, Watson and Crick had no idea about the bonding, that what type of bonding occurs in the rungs. The uprights are made up of sugar and phosphate, that's okay. But what is the arrangement of the bases in the inner region? And at first Watson and Crick imagined that the bases paired like with like. It was their opinion before observing the X-ray crystallographic image. They thought that the purines form bond with purines and the pyrimidines form bond with pyrimidines. And when they developed a structure on the basis of that assumption, then the structure didn't had this constant diameter because the points where the purine bases bonded had greater diameter than the points where the pyrimidine bases bonded. Then they reminded themselves once again about the Rosalind Franklin's X-ray data, where the diameter of the DNA was constant, uniform throughout its length. So they changed their mind and uh, they soon realized that the purines form bond with the pyrimidines. After considerable trial and error, Watson and Crick realized that the chemical structure of the bases dictated the pairs even more specifically. Watson and Crick's pairing scheme both fit that was known about the physical attribute and chemical bonding of DNA and explained some data obtained several years earlier by American biochemist Erwin Chargaff. Now, Erwin Chargaff in 1950s, in the early 1950s, uh, presented the Chargaff rule, in which he discussed the amount of different bases in DNA, and he said that the amount of adenine will always be equal to the amount of thymine, and the amount of cytosine will always be equal to the amount of guanine. Although the Watson-Crick base pairing rule dictates the allowable combination of nitrogenous bases that can form the rungs of the double helix, they place no restriction on the sequence of nucleotides along the length of a DNA strand. Means although the pairing in the inner region will be complementary, adenine always bonds with thymine, cytosine always bonds with guanine, but the sequence of nucleotides along the length did not had any restriction. It could be of any length and any arrangement, the nucleotide sequence. In 1953, it was 25th April, Watson and Crick rocked the scientific world with a succinct paper in the British Journal of Scientific Journal Nature that explained their molecular model for DNA. So they published this research paper on 25th April, and 25th April is celebrated as the World DNA Day. The journal where they published their paper was Nature. This is a prestigious journal, and the journal still exists. And one interesting thing is that Rosalind Franklin and Maurice Wilkins also published their work in the same edition in which Watson and Crick published their work, but the Watson and Crick paper received more fame, more attention from the community as compared to Rosalind Franklin and Maurice Wilkins paper, and it was only one page in length. The research paper which revealed the structure of DNA was only a single page, and that discovery is still one of the biggest discoveries, one of the biggest achievements in biology. In 1962, Watson, Crick, and Wilkins received the Nobel Prize for their work. Sadly, Rosalind Franklin died of multiple cancers in 1958 at the age of 38 and was thus ineligible for the prize. The prize was given in 1962 and 4 years before Rosalind Franklin died because of ovarian cancer, and Nobel Prize is not given posthumously means it cannot be given to a person after his death. So therefore, she didn't receive it. If she was alive at that time, then she could have been the fourth person. Few milestones in the history of biology have had as broad an impact as the discovery of double helix with its AT and CG base pairing. The Watson-Crick model gave new meaning to the words genes and chromosomes and to the chromosome theory of inheritance. With a complete picture of DNA, we can see that the genetic information in a chromosome must be encoded in the nucleotide sequence of the molecule. Now coming to the structure of RNA, we already compared a DNA nucleotide with an RNA nucleotide, and here is a RNA molecule alongside a double-stranded DNA molecule. The RNA molecule is made up of adenine, guanine, cytosine, and uracil nucleotides. Its sugar is ribose. It is short-lived because of that extra oxygen at position 2, which forms bond with other um, degrading agents, less stable because of that extra oxygen at position 2. It is single-stranded. DNA is single-stranded. DNA is double-stranded. But remember one thing, RNA can be double-stranded. Transfer RNA is double-stranded. When this RNA fold upon itself and the complementary bases come in front of each other, then they can form hydrogen bonds and the RNA can become double-stranded. But RNA is never helical. In RNA world hypothesis suggests that RNA acted as genetic material for early life on Earth. If you have a look at the evolution where the organisms have moved from a simple to more complex, so if you apply the same concept here, then RNA is simple as compared to DNA, and it can be regarded as an ancestor of DNA. These are the references from where the text and various pictures have been taken. Thank you very much. Allah Hafiz.